be looking now into aspects of molecular genetics. Now, primarily molecular genetics deals with A, genetic code, B, operon, C, protein synthesis, and we would be looking into various other dimensions of gene expression and gene regulation. Now, when you take genetic code, a genetic code is a sort of a program which any biological organism has. If you take the DNA is termed as a master molecule, or DNA is termed as a template molecule. The DNA has the nitrogen bases, and the nitrogen bases exists in the form of alphabets. Three successive nitrogen bases put together constitutes a codon, and a codon is specific for an amino acid. Hence, the language of the codons from the DNA is transferred into an anticodon into the mRNA or the messenger RNA, which are present within the nucleus and the nuclear membrane in a eukaryotic cell, or they are generally present in the region of the incipient nucleus in bacteria, and later on from the mRNA it is transcribed to the sort of ribosomes. Thereby, we have the information flow moving from the DNA to the RNA to the proteins. The most important concept number one is DNA, deoxyribonucleic acid, is always called the template molecule. When you say the template molecule, it means the master molecule, and it can code. The meaning of the term coding refers to the implication of DNA to protein relationship. When information on the DNA is converted to the protein, and we are talking about this sort of a transfer or relationship, it is predominantly termed as coding. So, during coding from the DNA, we have the nucleotides. The information which is present on the nucleotides via codons. Codons is a sequence of 3 successive nitrogen bases which are specific for any amino acid, or n number of amino acids constitutes a protein. Now, whenever we take a code is always a triplet code. Code is always a triplet code. It is made up of 3 successive nitrogen bases. A sequence of 3 successive nitrogen bases is called a triplet code. Now, each nucleotide specifies one amino acid. A single nucleotide is specific for a single amino acid. Now, when we look at the genetic code, we understand that in each nucleotide we have 4 nitrogen bases. Each nucleotide is predominantly constructed from 4 nitrogen bases, and if we have only 4 nitrogen bases, we could be, if they are very, very specific, if you think each nitrogen base is specific for an amino acid, then it could only be coding for 4 amino acids. However, that is, if you take 2 nucleotides, if you take 1 nitrogen base, we are looking at how many amino acids can get specified from 1 nitrogen base. 1 nitrogen base, if it specifies for 1 amino acid, then we would only be having the code for 4 amino acids, which is not true. If we say that 2 nitrogen bases or 2 nucleotides are specific for a single sort of an amino acid, then it would mean 4 to the power of 4, that is, it will be specific for 16 amino acids. 4 into 4 is 16 amino acids. Primarily, scientists have come with a 3-digit code, a 3-digit codon. A 3-digit codon means 4 to the power of 3. 4 nitrogen bases to the power of 3, thereby it is 4 into 4 into 4. From this, we can get 64 codons. 64 codons are allowable for coding 20 to 21 amino acids. Thereby, we understand that many triplets of codons can code for the same amino acid. They can code for the same amino acid. This is a very important concept. Now, there have been several evidences to state that the genetic code or the codon is triplet in nature. The codon is triplet in nature in biology. So, this is the most important principle. Why we have the triplet codon, how many codons are formed, and how does it specify for any particular amino acid. Now, we are looking at the properties of the genetic code. What are the properties of the genetic code? The first important property is it is triplet in nature. First, it is triplet in nature. That is, it is made up of 3 successive nitrogen bases. Second one is they are non-overlapping. That is, in case you have the nitrogen bases this way, one, 2 and 3 form a codon which is specific for an amino acid. Next, 4, 5 and 6 will specify amino acid 2. Whereas, there is never a case of 2, 3 and 4 constituting an amino acid. Non-overlapping. The code is degenerate. The code is degenerate. That is, you have about each amino acid can, with a single codon, can keep on coding for many, can keep on getting coded by different sort of codons. Now, if you take methionine, if methionine is taken and we have tryptophan, there are exceptions in that they are coded by a single codon. They are coded by a single codon. Single codon is seen in the case of methionine and tryptophan. The first of the nitrogen bases which can code, the code is according to codon rules. We have codon rules. As per the set rules, always A codes with C and G codes with C. A codes with U and G codes with C. But however, in a codon, when you take a codon, we have base 1, 2 and 3. The third base in any codon, the third base has the freedom and it can permit more than one type of a pairing at that particular position. So, if you take the third position, in the third position any other nitrogen base is allowed. So, because any other nitrogen base is allowed, this property is termed as Wobble hypothesis. So, the Wobble hypothesis indicates that in a defined codon, the third base is free to accept any other nitrogen base. Thereby, we have different amino acids whose codons are very specific as well as little bit flexible. Now, when we go to the characteristics or the features of the genetic code, the genetic code next one is commaless. It is commaless. When you say commaless, it means it is continuous. Continuous means non-interruptive. It is non-interruptive. And when you take the commaless genetic code, always we have poly A, that is AAA, which specifies lysine, L/S. Poly A specifies lysine. Poly C specifies proline. Then poly U and U is specific for phenylalanine. Phenylalanine, that is the amino acid. Another important property of the genetic code is it is non-ambiguous. That is, it is always very clear. There is a particular codon for a specific amino acid. There is a sort of a definite relationship between a single codon and a specific amino acid. And again, when you take in the properties, the genetic code is universal. Right from Escherichia coli to human beings, genetic code exists in all cells. So, the most important property of any biological cell is the presence of the genetic code. Now, the genetic code has been studied by Nirenberg and Mattei. Nirenberg and Mattei have worked on in vitro systems. When you say in vitro, it means test tube. So, they had synthesized a polypeptide, an artificial polypeptide had been synthesized in a test tube by using an artificial mRNA. So, both these scientists, Nirenberg and Mattei, had taken an artificial construct of a messenger RNA, a synthetic mRNA, and they had synthesized a polypeptide. And this method of synthesis of a polypeptide by using a poly UUU, they had introduced artificial uracil and uracil, and which codes for phenylalanine, and the experiment which they had conducted is also termed as the poly U experiment. So, thereby in a test tube you make a synthetic poly U. Similar is the case with relation to taking all poly A's as well as poly B's and poly C's, guanines, poly G's and polythiamines, which can specify an amino acid. So, this method of constructing an artificial polypeptide and synthesizing a polypeptide in a test tube led to the elucidation of the genetic code. Thereby, we exactly know for each a particular codon is specific for a sort of an amino acid. In a genetic code, always the starting codon is termed as the initiation codon, and the initiation codon is AUG. AUG is always called an initiation codon. The start of protein synthesis occurs due to the presence of AUG. And again, for a polypeptide to get formed, once you have an initiation codon, a polypeptide gets started up, and if the polypeptide has got to get started up in protein synthesis, the polypeptide should be formulated and it should be attached to methionine, F-met. And so F-met primarily is a starting point for a polypeptide. So for a polypeptide to get initiated at the ribosome and as well as at the protein level, you require the methionine which has to be formulated. Now if AUG is the initiation codon, then on the tRNA, the anticodon which is present on the tRNA is in the direction of 3' and it is an UAC anticodon with a 5'. So 3' UAC 5' is a sort of an anticodon which is present on the tRNA or on the soluble RNA. In addition to this, there are some codons which are termed as termination codons. Termination codons are called stop codons, or they are also called as nonsense codons. The normal codons are UAA. Next you have UAG, UAG, and again you have got UGA. UAA is called ochre. UAG is primarily called as amber. Is popularly called as amber. UGA is denoted as opal. Now, when you have a tRNA synthesizing proteins, when we encounter a sort of a termination codons, there is no tRNA synthesis, tRNA is not formed, but instead of tRNA, it codes for release factors. Release factors, that is RF1, RF2 and RF3. So tRNA is not formed, whereas release factors are formed from the sort of a termination codons. Now these particular nonsense codons can primarily sometimes also be mutated. They can be changed. So we understand primarily what is a genetic code, what is a codon, what are the characters of the genetic codon, what is an initiation codon, what do you understand by F-met, and what is a termination codon. Now next we go into the wobble hypothesis. Now what exactly is the meaning of the term wobble? Wobble indicates the specificity of the codon is due to the first two bases. When you are talking about codon specificity is due to base number 1 and also base number 2. Codon specificity is primarily due to the presence of base number 1 and base number 2. Thereby, the same tRNA can recognize more than one codon which differs on the third position. The third base is changed. If in the third base, in case it is altered, the third base if it is altered, the same tRNA can recognize the codons despite the fact that the third base is altered. So thereby here we understand in the third base the pairing is not stable, or the pairing is more open, especially with relation to the sort of a third base. Now many codons can tolerate mutations in the third base. In the third base, if there is mutations, one base gets substituted by the other base. Thereby, the DNA is able to be more stable. So, hence the wobble hypothesis states whenever there is a mutation, even though the third base is altered, the alignment of the proteins tend to remain the same. If the first and second bases are the same, the third position with different bases will code for the same amino acid. That is, you have 3 bases, despite the third base being changed, even though it is altered, it will always code for the same amino acid. It will always code for the same amino acid. So there can be substitution of the third base. Thereby, there can be accommodation of any other nitrogen base. So wobble hypothesis is a method for accommodating. There is accommodation of a third base due to it can be because of mutations, or in case there is any sort of an error, the error can be corrected, and even because of correction, the same amino acid is always read in a sort of a protein. Hence, the wobble hypothesis gives room for third base accommodation, adjustments for mutations and errors, corrections of the third base, thereby the same amino acid is quoted. Now, we have finished off on genetic code, basic concepts on genetic code, as well as the wobble hypothesis. We next go to the transfer RNA. Basic features of the transfer RNA. The transfer RNA is also called as soluble RNA. It has been studied by Hawley. Hawley was a scientist who discovered tRNA. It has a model which resembles a leaf, especially the secondary structure of the tRNA resembles a leaf, and it is termed as the clover leaf model. So the clover leaf model relates to the secondary structure. Primary structure of a molecule is just like a paper. The secondary structure is a paper which is folded, and if the fold is again transferred, it is termed as a sort of a tertiary structure. So normally the clover leaf model represents the secondary structure of the tRNA. And when you take the tRNA, it has got 3 loops. It has 3 loops. It has got a DHU loop. It has a DHU loop. Then it has an anticodon loop. And also a thymine loop. It has an amino acid acceptor arm, where the amino acid comes in joins. Amino acid acceptor arm, and this particular region is called the 3OH-CCA end. It is called a CCA end. The anticodon loop always has GAA. The DHU loop is present on the left hand side towards the 5' end, and again there is an extra loop between thymidine and anticodon loop, in case we have the anticodon loop here. There is an extra loop over here. Hence, if you take a tRNA, it resembles this way, and the correct alignment of the amino acid is due to the presence of this particular molecule of the tRNA, which is also termed as an adapter molecule. It is termed as an adapter molecule because just like a switch plug, it can take in the DHU end. We have the amino acid anticodon loop, extra loop, thymidine loop, and then we have the 3' and the 5' ends on this side. So this is in brief with relation to the sort of a tRNA. Now we are going to the charging of the tRNA. The amino acid, incoming amino acid in the cytoplasm, attaches to a specific tRNA, and it attaches to the specific tRNA in the presence of an enzyme called amino acyl tRNA synthetase. Due to the presence of amino acyl tRNA synthetase, an amino acid can attach to a specific tRNA. Now, one enzyme charges all the tRNA. This is the enzyme which charges all the tRNA that carries the same amino acid. So, always when you have a tRNA and it should carry a sort of an amino acid, it is charged by the enzyme called amino acyl tRNA synthetase. Now, when it gets charged, it results in the formation of an amino acyl tRNA. It results in the formation of an amino acyl tRNA. The anticodon in the tRNA, specifies the alignment of the codons in the mRNA. If you have got mRNA, the mRNA has got codons. These particular codons are recognized by the anticodon which is present on the tRNA. So, what dictates the anticodon on the tRNA dictates the proper alignment of the codons. So, recognition of the codons is the function of the tRNA. What is the prime function of the tRNA? It helps in recognition of codons. Now, where are the codons found? The codons are present on the mRNA. Anticodons which are present here base pairs with the mRNA codons. So, the anticodon-codon combination occurs. So, the recognition for specific tRNA for an amino acid attachment is due to the presence of amino acyl synthetase. Next, we are going into cracking of the genetic code. How do people know exactly what the code is made up of? How do they discover that the genetic code is very ambiguous? And again, how it is universal in all living organisms? When we take Nirenberg and Mattei, (1961), primarily they had synthesized an artificial mRNA. So, they are responsible for the synthesis of mRNA in vitro. When you say in vitro, it means artificial. In the test tube. So, like if you take a molecule of one base, like say U, we understand a poly-U uracil can be synthesized in a cell-free system and it can code for phenylalanine. If you take in all the enzymes and the cofactors which are responsible for protein synthesis, put them in a test tube, and it is a cell-free system, and in the cell-free system you are introducing poly-U. When you introduce poly-U, what you primarily get is phenylalanine. So the artificial synthesis of an mRNA, messenger RNA, you are loading an mRNA and you're getting a phenylalanine. This was the method by means of which they could exactly know that a particular sort of a codon is responsible for a specific sort of an amino acid. So this experiment is termed as cracking of the genetic code, which has been studied by Nirenberg and Mattei. That is, the experiment of Nirenberg and Mattei went like this. They had added 20 amino acids along with ATP and also guanosine triphosphate, and thereby they found each time an amino acid was added into a sort of a test tube in a cell-free system, they always found that U is specific for phenylalanine. Thereby they could synthesize a chain which is termed as a homopolymer chain. A homopolymer chain, U continuously phenylalanine. The synthesis of an artificial mRNA with a repeated sequence of known nucleotides was the pioneering experience and the experiment conducted by Corona. So Corona is attributable for artificial mRNA repeated synthesis of a repeated sequence of that and of known nucleotides, especially in the test tube. Thereby Nirenberg and Mattei as well as Korenberg were awarded the Nobel Prize with relation to codon specificity, mRNA, and help with relation to protein synthesis. DNA replication is a process wherein a DNA synthesizes the DNA. DNA synthesis is primarily termed as DNA replication. So during DNA replication, there is the presence of a replication fork. That is, if you have a strand of the DNA, the strand of the DNA at places where it has got to replicate will go to form a Y-shaped structure. The Y-shaped structure is termed as the replication fork. During DNA replication, the characteristic feature is it is semi-conservative. The meaning of the term semi-conservative is we have a DNA, we have a parental strand. The parental strand acts like a template, and when the parental strand unwinds, if you think in terms of a DNA molecule as being made up of a red and a blue ribbon, and the red and the blue ribbon unwind, the red ribbon in turn will synthesize another daughter strand which is called a rose ribbon, and the blue ribbon will synthesize another daughter strand which is a light blue ribbon. So always when daughter DNAs are getting formed, they will have one copy of the original DNA, parental DNA, and one copy will be that of the daughter strand. This pattern of inheritance is termed as semi-conservative replication. So in a semi-conservative replication, there will always be one original, and again there will be one daughter strand during the formation. When you take DNA replication, there are several enzymes which are involved in DNA replication. The first of the enzymes is termed as helicase. Now, if you take helicase, helicase is an enzyme which normally can unwind. It unwinds the DNA at the point of the replication fork. At the replication fork, the helicase can unwind the DNA, and when it unwinds, we have the formation of free nitrogen bases. The free nitrogen bases are free because the hydrogen bonds are broken. Then, in addition to the helicase, we also have SSB proteins, single strand binding proteins, which will come, and primarily they are the stabilizing proteins. They will see to that the nitrogen bases are not left off. Now primarily when the double helix, this is the double stranded DNA. The double stranded DNA in the presence of a helicase will unwind. So when it unwinds, there is a sort of a torsion which is built up on this side, on the left hand side. So to relieve the torsion, there is another enzyme. There is another enzyme called gyrase. Gyrase is also termed as topoisomerase. Gyrase is also called as topoisomerase 2, topoisomerase or topoisomerase 2, and it catalyzes the breakage and the reunion. So the objective of the torsion of the gyrase is to remove any sort of a torsion. It is just like two strings put together. When you pull the strings, you have it spinning on the anti-loop. So the anti-loop of the DNA is primarily controlled by the gyrase enzyme. Helicase is an unwinding enzyme, and SSB is a stabilizing enzyme. During DNA replication, there is presence of an enzyme helicase which unwinds the DNA. Gyrase is the second enzyme which removes the torsion out of the DNA strand. Out of the DNA strand, it removes the torsion. We encounter a replication fork, which is a Y-shaped structure when the DNA replicates or unwinds. Now, during DNA replication again, there is the presence of a small primer called RNA primer. The RNA primer attaches to the free nitrogen bases. RNA primer attaches to the free nitrogen bases and it synthesizes short primers. It synthesizes naked nitrogen bases which are termed as short primers. So a primer is a sort of a start signal. Now in the presence of another enzyme called DNA polymerase. Now DNA polymerase 2 is normally present in Escherichia coli. In E. coli, DNA polymerase 2 is present. So here it is, you got DNA pol 1 and pol 2 as present in prokaryotes as well as in eukaryotes. Now this DNA polymerase primarily starts DNA replication. It comes in and adds to the RNA primer. And it rolls on the RNA primer. So while it rolls, there is elongation, that is, all the nucleotides are stitched together. So there is elongation of the nucleotides. There is elongation of the nucleotides, as well as it does lot of proofreading. That is, the DNA polymerase edits the nitrogen bases. Thereby, any sort of an errors in the nitrogen base, if there are any errors in the nitrogen base, they are removed. Just like an eraser, the DNA polymerase runs on with the help of the primer. It tacks the primer like a sort of a zip, and while it keeps on pedaling up, it can remove up any of the free nucleotides or error nucleotides. Thereby, correction of the error, proofreading, and replication. Editing, replication, and proofreading are the three important properties of the DNA polymerase, which is a very important enzyme for during DNA replication. Now, DNA polymerase always travels from the 3' to the 5' direction. That is, when you take a DNA polymerase, when we have the strands, one of the strands which grows continuously is termed as the lead strand. On the lead strand, the DNA polymerase always keeps on going continuously. From the 3' to the 5' direction, it keeps on going this way. And sometimes we have another enzyme called DNA endonucleases. The DNA endonucleases breaks the bonds between the DNA. The DNA endonucleases can break the bond between the sort of a DNA. So thereby it generates free nucleotides. It generates free nucleotides. In addition to this, we have another group of enzymes which are termed as DNA ligases. Now, DNA ligases act like gums. They join up the sort of free nitrogen bases and the free sort of a nucleotides. So, during DNA replication, we have a replication fork. The strand which is synthesized on the outside is termed as the leading strand. The leading strand is continuous. It is formed first. It is called the leader sequence. And it is also formed fast in the presence of an enzyme called DNA polymerase. For the leading strand to get synthesized from the replication fork, as we understand, we have to have the sort of a DNA polymerase. And for the polymerase to start, we require a sort of a primer. Parallel to DNA replication, on the inside, we have the synthesis of short pieces of DNA. The short pieces of DNA are termed as Okazaki fragments. Okazaki fragments are small pieces of the DNA, and they keep synthesized in the opposite direction. And when you have the Okazaki fragments, they are joined by the sort of a ligase. They are joined by the ligase, and a new daughter strand is synthesized. Next. So, if we want to summarize on DNA replication, there is DNA initiation, that is DNA initiation occurs by strand separation, then there is unwinding, separation of the DNA, denaturation is separation, breaking of the hydrogen bonds, and there is the formation of one continuous strand. The continuous strand is called the leader sequence, and another small strand which gets formed is termed as the lagging strand. It is termed as a lagging strand. So, thereby the lagging strand is discontinuous. It is discontinuous, formed slow, formed on the inner side of the bubble. So, this area is termed as the replication bubble, and this one is termed as the replication fork. So, the replication fork primarily gets formed, it forms from the right as well as it also gets formed on the left. When you take a typical DNA which is undergoing replication, it will have lot of replication forks. So, replication forks are made up of bubbles, and the rule is complementary base pairing. So, primarily DNA polymerase is an enzyme which is involved in both proofreading as well as elongation, and lagging strands are short fragments. Lagging strands are short fragments of the DNA, and they primarily go to form the Okazaki fragments. Now, in the case of the eukaryotic chromosomes, if you take the eukaryotic DNA has several ORIs. ORI refers to origins. Thereby, DNA replication in a eukaryote can occur bidirectional from both the sides due to the presence of ORIs. These are all the sites of the ORIs. Whereas, when you take a prokaryote, in a prokaryote there is a single ORI. And similarly, even in the case of Okazaki fragments, when you come to the Okazaki fragments, they require several RNA primers. N number of RNA primers are required because every time a small piece has to be attached. So, these are N number of RNA primers. Whereas, when you take the leading strand, only one primer is involved with relation to the formation of the strand. So, if you want to summarize out, if you take the leading strand is formed continuously. There is no gap. Leading strand is formed continuously, and it is called a continuous strand. It is formed, and the DNA ligase is not required over here. It is formed from the 5' to the 3' direction. A single RNA primer is needed for the formation of the leading strand, and it begins immediately. Whereas, when you take the lagging strand of the Okazaki fragments are discontinuous, and DNA ligase joins the Okazaki fragments, and it is formed from the 3' to the 5' direction, and many primers are needed over here. Its formation is slow, and the lagging strand begins late during DNA replication. Next, we go to the operon concept. The operon concept has been proposed by Jacob and Monod. (1961). Operon refers to a cluster of adjacent genes which consists of a common operator and a promoter in the case of Escherichia coli. In Escherichia coli, we have a series of 3 structural genes . The 3 structural genes transcribe, they go to form an mRNA, and they get transcribed from a polycystron. So it is termed as a polycystronic mRNA, and they go to form an enzyme. An enzyme is made up of an amino acid. An enzyme is coded by the mRNA. The messenger RNA has got several functional genes. A cluster of functional genes is termed as a polycystron, and a polycystron is made up of structural genes, and the structural genes are 1, 2 and 3 in number. So the operon concept primarily establishes that genes are transcribable units. Genes are transcribing units. That is, they are capable of forming the mRNA as well as the enzyme. Now, in an operon concept, we have 3 structural genes. Then we have a promoter. And also we have an operator. If we take an operon is made up of a promoter, it is made up of an operator, and it is made up of 3 structural genes. A cluster of a promoter and an operator and 3 structural genes is termed as an operon. A promoter is a specific sequence of the DNA. It is a specific sequence which is bound by an enzyme called RNA polymerase. And RNA polymerase will come and bind to the sort of a promoter. It can bind to the sort of a promoter. And an operator is a short DNA sequence. It is a short DNA sequence to which is adjacent to the promoter, to which a repressor can come and bind to the operator. So when the repressor protein binds to the operator, it controls transcription. So the controlling element of the operon is the repressor. Now when we have 3 structural genes, we have 3 genes, gene 1, gene 2 and gene 3. The 3 genes along with the promoter and the operator operate synchronously. So if a gene goes to form the mRNA, it goes to form the mRNA, and it goes to form the enzyme. This pattern is primarily termed as gene expression. It is termed as gene expression. Now, gene expression primarily can be turned on or it can be turned off. It can be turned on or off. When we add lactose to the medium, if lactose in milk is added to a medium in which we are growing Escherichia coli, when lactose is added, there is the synthesis of 3 enzymes. 3 enzymes get transcribed. They get transcribed from the mRNA. And the 3 enzymes which get transcribed are galactourinase, permease and transacetylase. These are the 3 enzymes which get formed in the presence of lactose. Now, when there is absence of lactose, if lactose is absent, then the synthesis of these 3 enzymes is blocked. It is because a repressor binds to the operator and the coding or the gene expression of the 3 genes is off. Thereby, we understand that lactose can induce. Now, lactose present in Escherichia coli can primarily, lactose can start the sort of an engine. If lactose is absent, another protein called a repressor protein will bind to the operator gene and it will prevent the 3 genes from expressing to form galactose permease as well as acetylase. When genes are present in a series, we have a promoter and an operator and a cluster of genes. This continuity of genes is termed as contiguous. Continuity of genes is primarily termed as an operon. So, we understand operons or a cluster of genes can be both inducible, they can be inducible as well as they can also be repressible. Inducible means positive, repressible means negative. We are talking about lactose operon. A cluster of genes with a promoter gene, operator gene and 3 structural genes determine lactose metabolism. We understand lactose operon is an on mechanism. On means positive. That is, if lactose is present in the medium, lactose is present in E. coli. In the E. coli cells, we give lactose. Lactose can combine with the repressor protein. Lactose can combine with the repressor protein and also the operator protein and also the operator gene. So, we have a system wherein lactose combines with the repressor protein and it also strikes the operator. So, when it hits the operator gene, it prevents the binding of the operator. So, operator cannot bind. It prevents binding of the operator to the structural genes. So, a repressor primarily does not allow the operator to perform. Thereby, we have primarily an RNA polymerase, parallelly an RNA polymerase which binds to the promoter. It binds to the promoter. Binding of the RNA polymerase to the promoter is allowed. Lactose binds to a repressor protein and operator gene becomes non-functional. Thereby, we have the 3 structural genes. The 3 structural genes are LAC-Z, we have LAC-Z, LAC-Y and again LAC-A. These are the 3 important genes which are involved in lactose operon. So, the 3 enzymes primarily, they under the presence of RNA polymerase, because there is no binding, directly it can go and function in the expression of the 3 genes. Thereby, we have there is switching on of LAC-Z gene, LAC-Y gene and LAC-A gene, and thereby the LAC operon is on. And we have the formation of the enzymes. Now, sir, in certain cases, we have another enzyme called allolactose. The allolactose can bind to the LAC repressor. It can bind over here. So, when allolactose binds to the repressor, lactose is released, operator becomes functional and the entire process stops down. So, this is termed as allosteric effect. So an allosteric effect primarily reverses the situation. So during lac operon, the primary constant is lac operon is functional because RNA polymerase binds to the promoter. Promoter improves the structural genes. There is expression of lac Z, Y and A, and thereby the enzymes are formed. In an allosteric effect, allolactose binds to the repressor and thereby lactose gets released. Once lactose gets released, the entire function stops. When you take lac operon, that is lactose operon, is negatively inducible. The cluster of genes can function. We have a lac Z and a lac Y and a lac A. Now, when you say negative inducible, what is the meaning of the term negative inducible? There is a regulatory protein The regulatory protein is a repressor. The repressor binds to the DNA and transcription is, that is DNA making RNA is called transcription. Transcription is inhibited. The presence of allolactose inactivates the repressor. And allows the transcription. Thereby, it is positive. The structural genes are functional. Lac genes are formed. And the enzymes are synthesized. So, this process is primarily termed as negatively inducible. So, in negatively inducible, we have a regulatory protein, we have allolactose, we have binding to the DNA, transcription can be on or off, and again structural genes are functional. There is the expression of the lac genes and the formation of the enzymes. Now, we understand that if lactose is absent, the repressor binds to the operator. When the repressor binds to the operator, it prevents transcription. Due to binding. Of the repressor. After gene control, we go into tools of genetic engineering, and in the tools of genetic engineering we have plasmids. Now, plasmids are extra chromosomal elements. They are present away from the bacterial DNA. They are resident plasmids, and plasmids are small circular DNA molecules which can carry your foreign gene of insert and are used as tools in genetic engineering. So, what are the typical characters of plasmids? They are present in Escherichia coli. They have an origin of replication. They are double-stranded circular DNA molecules. This is an ORI, the origin of replication, and this indicates double-stranded DNA molecules. And whenever you take a plasmid has marker genes. it has a left flank and it has a right flank. It has left and the right flank. And any particular plasmid can easily be isolated. The features of plasmids are they can easily be isolated. They need to be small in size in terms of kilobase pairs of nitrogen bases. And they should be capable of integrating a foreign DNA should be capable of being integrated within the plasmid. So, if it is integrated, it is termed as an integrated plasmid. Integrative plasmid. So, primary plasmids are vehicles for genetic engineering, and normally when you take a plasmid, the plasmid will also have certain selective markers. The selective markers can glow in the dark because they normally have a green fluorescent dye. GUS, green fluorescent dye marker, is popular in plant genetic engineering. And when you take a plasmid is measured in terms of kilobase pairs. So, the basic attributes of any particular plasmid is it should have an ORI, it should be double stranded, foreign DNA should be integrated, it is normally present in Escherichia coli, it can easily be isolated, and it is small, and we can convert a sort of a circular plasmid into a linear plasmid. A circular plasmid can easily be converted to a linear plasmid during genetic engineering. The most popular type of plasmid is termed as PBr322. Now, PBr322 is an artificial plasmid. That is, it is synthesized in the lab. It has been discovered by Bolivar and Rodriguez. Hence, it is termed as abbreviated with the notation BR. P refers to the plasmid and it belongs to the series 322. So, when you take a plasmid normally is the nomenclature occurs in terms of the plasmid discoverers and the series number. So, PBr322 is a common gene cloning vector. Of E. coli. Its copy number is 15 to 20. The copy number of the plasmid is 15 to 20 per cell. If you take a bacteria, we have the bacterial DNA and we could have plasmids if this particular plasmid was a PBr322, it'll per cell you will have 15 to 20 copies of it. So, presence of the copies of it is termed as a copy number. A PBr322 is a circular double stranded DNA molecule. It is a double stranded DNA molecule which has got 4.3 kilobase pairs. KBP, that is 4,300 base pairs is present in this, and primarily it has got 528 restriction sites. So when you take a PBR 322, it has an ORI, it has a left flank, it has a right flank, and all along the right and the left flank there is the presence of marker genes. PBR 322 has got a marker, antibiotic resistant marker called AMP, ampicillin resistant, and again tetracycline resistant. These are the two important marker genes. So the two selective markers, they are termed as selective markers, are ampicillin and tetracycline resistant. Now within the tetracycline, within the ampicillin and the tetracycline markers, we have several sort of regions which are recognized by restriction endonucleases. Restriction endonucleases are enzymes which can cleave the plasmid at specific regions. Like common restriction endonucleases are BAM H1, as well as we have even ECO R1. Then again we have HIND3. They are a series of restriction enzymes. So these restriction enzymes can come and cut at the marker sites. They can cleave here. Thereby when it cleaves the sort of a plasmid, it is just like a bangle is broken. Now what are the advantages of PBR 322? The advantages of PBR 322 is, first advantage is it is small in size. Second thing is it is circular. Third, it is present in Escherichia coli. Fourth, it has got marker genes. And fifth, it is a basic plasmid. That is, to the basic plasmid, you can add an operator, or you can add a structural gene, or you can add a promoter gene, and you can construct. So it was the first artificial plasmid which has been used in genetic engineering, and it can be upgraded. So PBR 322 is a very important, basic, all-purpose, convenient plasmid used in genetic engineering. We go to the second plasmid, which is called as PU18. Plasmid developed from University of California, number 18. Now PU18 is again a double stranded DNA. It is a double stranded DNA and it has unique restriction sites. It has unique restriction sites. It has a site for ampicillin resistance. Ampicillin resistance, it is of the size of 2.7 kilobase pairs. It can be digested by a single cut. So it is termed as a single digest plasmid. And the circular DNA can easily be converted to a sort of a linear DNA. The biggest advantage of PUC18 is any recombinant DNA that is, a recombinant DNA is one in which the foreign DNA is integrated, and multiple copies of that can be made by a technique which is termed as gene cloning. So the recombinant DNA or the foreign DNA can easily be inserted into the PU18, and it can be integrated along with the same sort of a cut which occurs due to the presence of an endonuclease. So an endonuclease can make cuts in PU18. When you say cuts, it means nicks. Nicks means breaks. So it can break PU18 to create a space for the recombinant DNA to come in. And the advantage here is the PU18 has got a lag promoter. What are the advantages of PU8 when compared to PBr322? It has got a lag promoter. Then it is a polylinker. Polylinker means this is a plasmid which just acts like a sort of a lorry. It can take in a lot of genes on it. So it is a polylinker, it can take a cluster of genes on it. And it has got a lag Z promoter, and it can code for beta galactosidase. It can code for beta galactosidase. So due to the presence of a lag promoter and due to the presence of a polylinker, it is of big advantage for PU18. Plasmid called PUC18 can undergo hydrolysis, that is, it can break down in water, and it can liberate a gene called XGAL, the lag-gal gene. And whenever lag-gal gene is synthesized, it produces a blue dye in cultures. So lag-Z is minus, forms a sort of a blue dye in cultures, and thereby whenever we have the presence of PUC, it can go to form a sort of a blue colony in Escherichia coli. Sometimes the blue colonies in a medium is primarily a good indicator of PUC. Now if you have a recombinant DNA, a foreign DNA of insert which is carried by PUC18, when it's carried by PUC18, it primarily what happens is it goes to form white colonies. In culture. The reasons being, the white colonies indicate it is beta gal deficient. It is beta gal deficient. So especially if it is grown in an X gal medium. So the presence of white colonies indicates there is successful recombinant DNA getting transferred. This is a sort of a colony indicated test for PUC18.